They taste pretty gross. Actually, doesn't taste
Alright guys, let's talk about viruses. So we're going to talk about mainly what they're made of, if they're considered alive, and how they reproduce. They're not. Yeah. Spoiler alert, they're not considered alive. Okay. So, characteristics of viruses. So, you're asking yourself maybe, are they prokaryotic or eukaryotic? Because since now we put something in that bubble. Either they're prokaryotic like bacteria, or they're eukaryotic like animal cells, multicellular organisms, fungi, stuff like that. But viruses don't have a cell membrane, and they don't have organelles. So they're neither prokaryotic or eukaryotic. They're not really cells. So since they're not cells, they can't be unicellular, and they can't be multicellular, because they're just nucleic acids and proteins. Yeah. I don't know. It is kind of weird. And then one of the categories for life is that they have to make energy, they have to use it, and they don't make energy. And they don't really have the machinery to reproduce either. And we'll see that in a later slide. So they're neither autotrophic or heterotrophic. So they don't make their own energy, and they don't consume it. And so then, which kingdom do they belong to in the 6 kingdoms of life? Well, since they're not living, they don't belong to any kingdom. There's no virus kingdom, you know? They're just kind of there. There is a classification for them, which we'll go over in a second, but it's not like the other kingdoms. They don't have a kingdom. I mean, there is a classification, but it's not like the other living organisms. They don't have a kingdom or a phylum or a class or order. So like I said, the viruses are made of 2 parts. Proteins, which is called a capsid. So make sure you highlight or underline this, something to remember it, because it's important. And genetic material. So they have genetic material either in the form of DNA or RNA, so nucleic acids. And then some of them have an envelope. So this is just an extra layer of protection. It's just more proteins. So this is a little good graphic. So in order to be considered alive, you have to grow and develop. You have to obtain and use energy. And then you have to respond to environment. And if you look, you have to change over time. So like whenever you have DNA, it replicates and there's mutations, spontaneous mutations, and then therefore you change, like evolution. So if you look at this right here, viruses don't grow and develop, they do not obtain energy or use it, and they don't respond to the environment. They do change over time though. So whenever they're replicating, their DNA will form spontaneous mutations. And that's why you hear of swine flu, like from pigs, it mutates into humans. And the only way they can reproduce is with the host cell. So like I said, they're just proteins and nucleic acids. So they don't have any of the machinery. They don't have RNA polymerase. They don't have ribosomes. They don't have any of that stuff to make more of themselves. So then this is a good little graphic. The Venn diagram, 3 circles. Make sure you take a picture of it if you don't have access to Edmodo. It's a good review for the star. So not only does it talk about the differences and similarities of prokaryotic cells and eukaryotic, but it also includes viruses. So you can see that viruses have little to them, but they're really different from... The things that are different about them is that they have these proteins and the capsid and an envelope. But they do not have a nucleus, they don't have any organelles, and they have genetic material. And some of them have a little bit of cytoplasm in them. So examples of viruses. This is a bacteriophage. So they're specific to bacteria. They infect bacteria, as it might sound. And they're made up of genetic material, the viral DNA or RNA, a sheath. This is what allows it, when it attaches, it injects the DNA into the host cell. So these points of attachment, the tail fibers, it looks like this for bacteriophages, but if there is a circle virus with appendages sticking out, these are points of attachment. If it was a triangle, these are points of attachment. And even some of them are like a snake-like structure. This is where the capsid and the genetic information is at, but this is the point of attachment because it's an appendage. So any appendage you see is a point of attachment. What's that, Carl?
What's the name of this one?
Let me see.
Is that it?
What's the name?
Like that?
Oh yeah, an envelope.
No, this. Like, draw an example of what's bacteriophage.
Oh yeah. That's it. That's it. And then this is the protein coat, the capsid, right here. The capsid is what holds the genetic information. So how do viruses multiply? That's the question, right? So on its own, a virus will sit on the surface and it will just remain dormant. I mean, it won't infect anything, it won't multiply. It's just one little virus particle. And it doesn't need anything to live because it's not living. So it'll just sit there until it attaches and injects its DNA into a host cell. And then it goes one of two ways. So you see lytic, think of sick. Lytic, sick. So that's when it actively replicates. And then it'll burst open the cell and it'll make more. Lysogenic is more like what is it called? Cold sores. Whenever you have a cold sore, you don't always have a cold sore. It enters in this dormant phase for a while, and that's the lysogenic. So lytic is immediate, lysogenic, think of hibernating. So lytic, sick, lysogenic, dormant. Hibernation. What's up, Jack?
So are there like certain conditions that draw lysogenic cycle?
Yes, that's a good question. So Jack asked, is there certain conditions that draw lysogenic guys in front? Okay. It doesn't draw out lysogenic into being lytic, and yes, there is, but it's still being discovered. So sort of like HIV, you can have HIV for 10, 20 years, and it never enters into the lytic cycle. And the reason it enters in is still being studied. They don't really know why. So it can be like suppressed immune system, or it can be environmental changes, like environmental stimulant, stuff like that. But those are just hypotheses. So whether it enters into a lytic or lysogenic, the bacteria attaches with the appendages, those little tail fibers, and then it injects its DNA into the host. And this bacteriophage is just a fancy word for what, guys?
Wait, what?
A bacteriophage is just a fancy word for what, June?
It's a fancy word for, I don't even know. Bacterial?
A bacteriophage.
Oh, okay, I was right. Okay.
A virus.
A virus specific for bacteria. So it injects its DNA or RNA into the bacterium. It forms a circle, which is called a plasmid. And then it has two ways, remember? Lytic, sick. So lytic, they start to metabolize, use the bacteria's machinery to make more parts. So it'll go and make more parts of the protein legs, and then it'll make more parts of the capsid, and then it'll replicate its DNA using the RNA polymerase and stuff, or DNA polymerase and stuff like that. Then it's sort of like going to like McDonald's. You have like the patty already made, the buns, and then it'll assemble it together. Then after a while, it'll make so many that the cell will lyse, and that's just a fancy word for like explode.
Wow. Why?
So it'll release those particles out to attach to another cell. So whenever you have a cold, it enters into the lytic phase right away. And then if your throat is sore or something, it's because it's raw, and all the cells, the first layer of cells, have ruptured and made new. And then those, whenever it ruptures, it sends chemicals to your brain to receive pain. It's like, okay, something's going on here. You need to take care of it. Then the other phase is lysogenic. So lysogenic is hibernating. It's dormant. So remember, it attaches, it inserts its DNA, it makes a circular ring, but instead of starting to assemble itself, it'll insert itself into the bacteria chromosome. So its DNA will become part of the organism's DNA.
Oh, so it like merges?
So the virus that they had, right?
Yeah.
Is that lysogenic or what?
So it like goes to
Lytic. So the virus I now read about, it didn't have a dormant phase. As soon as it entered the host, it started to affect them, right? So it was lytic right away.
Then how can
A little bit. Because it happens right away, but it has to it lights your cells. So when it lights just one cell, you don't really feel the effects. But when it lyses 100 cells, it has to be starting to feel it. So it's like an exponential rate. So you get it, it takes a couple days, suddenly it's you know what I'm saying? Good job here, guys. They said if you get it right away and it starts to lyse your cells, then how come you don't feel the effects? Like, how come you don't know exactly when you get the virus? And that's because whenever it lyses one cell, you don't really feel the effects. It's when it lyses 10,000 cells is when you feel the effects.
That's long.
So for this one, the lysogenic, it enters its DNA into it, and then when it replicates, the DNA is still there. And that DNA that was part of the virus is called a prophage. So that section of DNA is called prophage, which is the bacteriophage's DNA. So if you think about it this way, it takes a bunch of cells to feel it. Well, you enter the lysogenic, it stays that way, it makes a bunch of cells, and as soon as it hits the lytic phase again, you'll feel it right away, because a lot of the cells will do it at once. So the bacteriophage, the prophage, will exit from the chromosome, and it will enter into the lytic phase. And there will be, multiple cells doing that at once, so you'll feel the effect right away, versus whenever you first get the virus, and then one cell lyses, and then those, 100 viral particles go and infect 100 different cells, and then that's an exponential rate.
Wait, doesn't the lysogenic then go to the?
Yes.
So it's 2 stages.
Yes.
Right? It's this first one and then the second one.
Yeah.
Oh. Gotcha.
But the lysogenic phase is a dormant phase, where it just has no effects for a while, like I talked about with HIV.
Mm-hmm. Gotcha.
So now, how are viruses classified? They're not classified the other way, with the kingdom, class, order, genus, species, but they are classified by shape. So this shape, the circle, the square, the bacteriophage's shape, and by what host they infect. So there might be some for insects, or plants, or bacteria, prokaryotic cells. So this is an example. It's influenza. So you see those little projections on the outside of the circle? What are those for? For attachment, correct. Now, what about... So influenza is airborne. So for most viruses, like the one you read about, you have to come in contact with bodily fluids in order to contract the virus. But this, you can just speak to somebody, and it could be in the air particles. So they don't use any energy to move, so they just float, basically. So if they're on a surface and you touch it, and then you put that to your mouth, then you contract the virus. But with the flu, I can just talk to you right now, and if I have the flu, you don't have it.
We're done.
Whoa.
Hey.
Now, can I ask?
Hey.
No.
No. And then HIV. So this is contracted in a different way, but it stays in the lysogenic phase for X amount of time, and it depends on the person itself. So some people can have it for 21 years. It stays in the lysogenic phase. Some people can just die with it in the lysogenic phase.
Uh-oh. That's not bueno.
Well, not die from HIV, but die by old age.
Oh.
But something may trigger it, an outside force, environment, a suppressed immune system, stuff like that, and it'll enter into the lytic phase. So smallpox, that was completely eradicated from the human population with vaccines.
It's like
It's like extinct.
Aw.
No. Okay, and this is the Marburg
No.
virus. That's how it looks.
That's how it looks?
Yep.
So it's like
like a worm.
So another thing to think about when you're looking at these viruses is you saw the bacteria phage on top of the cell, the bacteria, right? And think about how small the bacteria cell is. Guys, listen. Think about how small the bacteria cell is compared to a human cell, and think about how small that bacteriophage was and compared to the bacterium.
Yeah.
So they're super tiny.
They're like this little.
This is 30,000 times magnified.
And you have like trillions of those in your body.
And there's
Well, if you have trillions of those, you would be.
Exactly.
you'd be dead.
Okay. So this is Ebola.
Yay!
That looks like a shoe.
No, yay.
Mickey Mouse.
So remember, they're classified by what they affect, who they affect, the host, and their shape, and their function. So that's why Ebola and Marburg are closely related. So 3 types of hosts. There's plants, animals, which include insects and stuff like that, guys,
Foot and mouth disease?
and humans.
What's a foot and mouth disease?
It's when your foot and your mouth switch places.
So treatment of viral diseases.
I got a question. Is chickenpox just like smallpox?
No, chickenpox is not like smallpox.
See, how would I know because smallpox is the same?
Yeah, smallpox kill people. Chickenpox, they can kill you, but they probably won't.
One time I thought I got chicken pox, but it was like really allergic to this one shit. I had like hives all over the back of
Oh
me.
gosh. Yeah, chickenpox usually originate in your stomach and then spread out.
It felt so weird.
What's
I
up?
felt like I had a bunch of
Hold on, guys. Shh, Jack has a question. Shh. What's up? I'm not sure, actually. I can look them up. Or just drop this over.
All over you, like sores, really bad sores.
Overseas thing.
Fever,
Looks like that.
What in the world? Looks like a robot.
It
Cut
does.
away.
much more.
There's a picture up there, but that's kind of
I think
weird.
that's bacteria.
Cover the body.
But, yeah.
And leave sometimes.
Because Jesus
Yeah.
cured the lepers. 12 lepers. And only one was thankful.
I'm thankful. Sure. Alright, guys, so viruses can't be cured by antibiotics because antibiotics will destroy a certain structure of bacteria's like cell wall. It's like peptidoglycan, that's what it's called. But it'll destroy that basically. But viruses are just protein and DNA. So we have protein and DNA in our cells, so it's kind of hard to destroy a viral particle without destroying your own cell. So like I was talking about, whenever your cell lyses, and then I said that those signals get sent to your brain to let you know that something's going on. Well, whenever you take medicine for viruses, it's usually alleviating those symptoms. So whenever you take cold medicine, because you have a fever or something like that, it's usually just alleviating the symptom of pain, stopping those signals to your brain. So antiviral drugs, there's some drugs out there that'll stop the attachment of the protein, the viral legs basically, to a host cell.
Sorry.
So they don't destroy the actual virus, like I said, because it's sort of hard to destroy a virus particle without destroying your own cell. viruses. So yes, they can affect us, and yes, they're bad if they infect us, but we can also use them to our advantage. So vaccines. Vaccines work by mashing up. So for smallpox, that was completely eradicated. We took the viral particle, we mashed it up basically, so it's just bits of it. It's not the whole thing intact, because if it was, it would infect you and you die. So you just put the DNA part in there, and then you put the protein part in there, and it's all like blended up, and then they inject you with it. So your T cells will go, get a little bit of it, and then be able to recognize it whenever they see it later on, and then say, hey, what's that guy doing in there? Kill it.
So you're telling me when you get a flu vaccine, you get the flu put in you, that way that you can
Yeah,
kill it
it's
yourself?
Yeah, so it's mashed up, like I said. They just cut it up and then inject it. No, it's not. So it's like if I took this cell, right, and I said, I got like a knife or something and just cut it all up. So then you would just have this little appendage, a little bit of the side of the cell wall, and then some DNA. But you wouldn't have this in you.
I don't like that still.
Not the whole thing.
I don't like it. Where do they even get it from?
It wouldn't go back
Do
to
they
this.
get it from like another person, or do they just find it in the air? Or like?
You can synthesize it, and we have technology enough to synthesize proteins and stuff like that. Okay, so I can show you an example where we can synthesize it. So vaccines, another thing is genetic engineering. So engineering something that you want. So if you wanted, you could put a bit of DNA into a prophage, and then it can put into a bacteria cell, and it can make whatever you want. You can make that virus and then cut it up, basically. Or another good example is diet ice cream. So diet ice cream is mainly water
I bet you.
because you want lower calories. So what they do is there's this Arctic fish that has a gene in it that will have like antifreeze basically. Genes. So they take that bit of gene, they slice it, they put it in a bacteriophage that we saw, that robot-looking thing. And then that phage, they'll put it to a bacteria, and it'll enter into that lysogenic phase, where it'll have that bit of chromosome inside, it'll have that bit of DNA into its chromosome. So now that bacteria possesses a gene that can make protein so it can have antifreeze properties. So they put that bacteria into the ice cream so that way it's not crunchy like ice and it's creamy like ice cream. Ooh. What's up?
So we're putting fish genes into food?
We're putting fish genes into bacteria and putting those bacteria into food.
All right.
The powers of modern science. And then agriculture. So we can use viruses that only affect insects instead of harmful toxic pollutions like DDP, which was used in the 70s, and then it's still showing up in our lakes and stuff. It's really toxic. Okay. Is it?
Click to stop the recording. Sorry. There. That one, yeah.